Thank you very much, Eduardo, and thank you to all the organizers for inviting me. And it's a pity, of course, that we cannot meet in person, and because in science, as in whole life, human interactions are the basis of our motivation. But it's a tough time, but we look forward that things will be better. So there is a minor technical issue now that I cannot give the presentation. I cannot drive, but Eduardo has to activate, has to press the slideshow. So please, I apologize if I say too often the word next or whatever. Yeah. But I hope you all understand me well. I don't see any protests, so I just continue. And my talk will be about soil microbial communities, and I will give a relatively general introduction to a few things, and then I get more and more specific to work that we have done or that we are actively doing in my research group now. So what we know about soil microbial communities, just to start with, as I am not sure about your specific background in microbiology, just give you a few hints. So what we know is that soil microbial communities are highly diverse, metabolically very versatile. They can do a lot of different things with their transform different compounds. They live in huge numbers in soil. But on the other hand, for example, in 1 gram of soil, we estimate there are as many bacterial cells as there are human beings on our planet. So it's a lot of high numbers, but still, when you consider the enormous surface area of soil particles within a gram and calculate, and this is my own calculation, so I'm not sure, I never heard big objections to this number. I calculate that only about 0.1% of the soil particle surface areas are covered with bacterial cells, which means the bacteria live a pretty lonesome life in soil. And what do? Nevertheless, they transform energy and substrate. So they don't do the metabolism to please us, but they do it because there is energy in the system of soil and there are substrates, and organisms take advantage of that. But for the good side for us is that this translates to ecosystem services, which means that they do something that is good for the ecosystem. And I come back to that a little bit later. So the transformations that the microorganisms in soil do, they are controlled by biological and non-biological interactions. And the biological interaction is very important. There are factors like competition, so organisms compete with each other or they cooperate with each other. And then the abiotic interactions are very important because of sorption and desorption of substrates, which provide this energy to the system. So this interplay between biotic and abiotic interactions generates community dynamics in space and in time. And I come back to that later. So that means that the environmental conditions affect the microbial community structure, and at the same time, the microbial community structure changes the environmental conditions. For example, a pH can decrease because organic acids are metabolites and so forth. So there is an interaction between these factors, and it's not very easy to separate these things from each other. Next slide. Yes. So just to remind you of what's going on in soil, there's a very different groups of organisms which live in soil. And here you see in the upper row the examples of bacteria, archaea. These both are prokaryotes. Then you have the fungi, different types of fungi, and you have the protists, which is a big group where we don't know a lot about still, especially in regard to their contribution to microbial activity. And then in addition, you have now, of course, no talk without viruses. So and it's really true in soil. You have a lot of viruses. And here I show you only 2 examples. This is on the left bottom. You see the bacteriophages, so the viruses which control or destroy bacterial cells. And next to it, you will see double-stranded RNA microviruses, so viruses that infect fungi, and both have a strong effect which we really don't understand well on the community interaction so forth. So next one. And you see this is just wants to symbolize with this image that there is a very, very complex interaction within each of these groups, because there are many different taxa species within a group, but also between these groups, which is still a major challenge. Yes, next one. And together, so they provide these ecosystem services. Here's a nice recent review about this from Italy also. And so I don't want to go into the details of this ecosystem services, but since you are in your school discussing this intensification of agriculture, but in an environmentally friendly way, you could call it ecological intensification. This means you have more intelligent ways to do agriculture and still get a good yield without the concepts of what I would call industrial agriculture, and with more knowledge than even, or with modifying knowledge from traditional agriculture. But if we want to do it, next one, we see that this requires knowledge about the organisms that provide these ecosystem services, about their regulation and their metabolism. And it requires an understanding of the soil habitat conditions and variation. So it's not possible to study it by just observing bacteria on an agar plate, but we need to understand what they do in their habitat. And also we need to learn how to upscale information. We see, let's say on the microbial level, we see a process, but what does it mean for the whole ecosystem? There is still a lot to learn. Okay, the next one. And just to remind you of what soils are made of, I show you the simple building blocks, the primary building blocks of soil, and these are the primary particles, and they are differentiated according to size, clay, silt, sand, and also particulate organic matter. And of course, the proportion of these defines the soil structure. And you see, like for example, the sand and the minerals you see on the bottom of the slide, and to the right, and the clay, they have different minerals. And that also means they have different absorptive capacities to release nutrients, to absorb nutrients. And what I show you within the next couple of slides from our own research, it also has different affinities to be attached to bacterial or fungal taxa. So to study these particle sizes, you can separate them by dry sieving or wet sieving. And in our own research, we did this wet sieving, which is based on suspending soils and then ultrasonicate them so that the particles separate from each other. Then you sieve them and centrifuge them. That's a common approach to study, for example, the organic substrate that is associated with particle size. But we have modified a protocol some years ago, and we reduced the ultrasonication energy so that we could keep the microbial cells attached to the particle size fractions. And so when you do that, you can really separate the soil into these fractions, and we photograph them. This is from one soil, and you see the clay, fine silt, coarse silt, sand, and POM, and you see they are very different already by just looking at them. Like the clay is much darker because it has stronger absorption and complex absorption of organic material, and so forth. And now we have, we were able to, quantified and sequenced the microbial communities that we found with each of these fractions. And it was very clear that they were very distinct from each other. And we could show that bacterial taxa, for example, of the group of Planctomycetales, which are highly abundant in many soils, they love to be associated with clay mainly, with the clay particles. They have a preference with clay particles. Whereas in the fine silt, we find actinobacteria and nitrospira, and in coarse celled Germanimoradites, and in the sand POM fraction, which we did not separate, so POM means particular organic matter, we have Bacteroidetes and alpha proteobacteria. So this is interesting because it shows that the bacterial taxa, they have evolved in different kind of niches where they attach to, and it makes sense because depending on the absorption and desorption, the nutrient concentrations will be different at these particles. And surprisingly, we also found this for many fungal taxa that they showed this preference. Here you see a heat map, which is I don't like heat maps very much personally. Other people in my field like it a lot. I think they're a bit, but they can give you a first idea of differences. For example, you see that in the upper on the left side, you see this block with the sand POM, and you see there is there's a group that does not occur as such in other groups, and you can you can sequence also that, and you see now on the next slide again these particle size fractions. And there you see that, for example, for clay, we found many more Penicillium and Mucoraceae and less Sordariales, whereas in fine celled we found Sordariomycetes, and in coarse celled we found also some taxa, but they always also had a preference for the sand POM fraction. So it's not always super exclusive that one fungus prefers one of the particle size fractions, but it can be an overlap. And also, on the other hand, if we go back, please, on the other hand, you see that even the sand POM, there are also some that avoid some fraction. For example, the Mortierella, they are evenly distributed in clay, fine celled, and coarse celled, but we never see them with sand and POM. Yeah, of course, all these names, they should not confuse you too much, and you don't need to remember those. And they may even be different in other types of soils, but it shows you that there is, in general, there is some interaction between the mineral surface or the clay, the particles size fractions, and specific taxa. And this makes sense if you think that the microbial communities evolved over 100 millions of years. It makes sense that they don't all are homogeneously distributed, but they are attached to different surface types. And this could be shown with our work quite conclusively. And you see up there are indications for the publications. So in general, if you have questions about more details, I am happy to provide you the publications as well, or Eduardo can provide them, and I can send them to Eduardo. So now I want to make a switch in my talk and talk about, so all the data you saw, they are a result of DNA analysis and the DNA that you can directly extract from a soil sample, right? And the way to do that, this is a protocol. The first protocols have been developed already in the end of 1980s. And since then, first it was phenylchloroform methods, but now over the last 15, 20 years, these kits became very popular to use. And some companies are very successful. Here you see a couple of these companies to do this DNA extraction kit. And everybody uses them for their soil microbial community analysis. And this is also good for several reasons. The first is you don't use a lot of phenol, which you normally use for the classical method, so you generate less waste. Probably you generate a bit more plastic waste though. But then you have this, you have a standardized protocol. So if you do a certain protocol of a commercial company, you can be pretty sure that somebody at the other end of the world who uses the same protocol gets comparable results, and not each lab has its own protocol. So that makes sense. But what is the disadvantage of this? And the disadvantage is that nobody thinks about why. why to use it. Yeah, typically you take one of the kits that your boss recommends, or maybe even you have a bachelor or master student who compared the kits, and then you take one of them. And they all have something in common. They extract total DNA from about 250 milligrams to 500 milligrams of soil. Then they do bit beating, so to crack open the soil, the cells, and then purify the DNA. Then you quantify the communities by quantitative PCR. Normally you do real-time PCR, or you sequence the PCR amplified genes. And for example, 16S RNA genes for bacteria, archaea, or 18S or 18S for bacteria. Then you group the sequences, you get into operational taxonomical units by 97%. It's a very rigid value. And then you say, okay, that is something close to the species level. Or more recently and more accurately are these so-called sequence variants, which are now used. They don't have the rigid 97% number, but they are a bit more flexible and consider other factors which are closer to the species level. And then you do phylogenetic assignment. So, and then followed by that, you can now say, okay, what is the diversity in my sample? That is what we call the alpha diversity. So basically, which taxa occur in my sample? And then you compare your samples. For example, you have one from a fertilized field and one from a non-fertilized field. Then you compare, and then you see the differences. And then you try to explain the differences, and you correlate this with soil properties you measure. And now this is a critical point, which you will see later in my talk. Which parameters do you use to characterize the soil property? Basically, what you do is, in most studies, you select the soil properties which are easy to measure and cheap. Yeah, that's the practical answer. Yeah. So everybody measures the pH. Yeah, it's easy. You make it suspension, or you think it's easy. Actually, it's very difficult to measure pH of a soil, but you can just make a suspension and measure, and you get a value. Then you determine total carbon, total nitrogen, etc. And I come back to that a little bit later. And then basically you speculate on the functions, because normally with 16S genes or ITS sequences, you don't know a lot about functions. There are in silico tools like PyCrust, but they are very limited in their capacity. And then you do similarity multivariate analysis. But what now? Okay, let's say you describe a community in this way, but what have you actually measured? That is the question. And basically, the community you look at is a result of what we call habitat filtering. Yeah, and that means it's a result of a selection process as imposed by the compositional characteristics of the soil and the history of that particular sample. For example, the compositional characteristic is the mineralogy, the organic material, the type of plants that were cultivated on that, and then the history is, for example, the agricultural treatment and so forth. That means you basically look at a community that has been selected on by different factors, and the temporal scale for the selective factors acting, they range from hundreds of years, if not more, if you think about the mineralogy of a soil, to hours. Yeah, let's say you take a sample shortly after a farmer spread a manure on the soil, then it would totally mess up your community analysis. You would see something that is a result of just what happened hours ago. Yeah, and everybody knows that, but it has to be care has to be taken how to interpret the results. Yeah, how do you want to say that a specific taxon is there because of the mineralogy or because of the agricultural treatment? It's very difficult to do that. But there is another problem. Because of your DNA extraction method, everything, you look always at the community at a gram scale. Yeah, your unit is 0.5 gram of soil, because that's where you got the DNA extracted from. So that is also something you can think about. Is that really what I want to? Is that the most important scale? Now in the following part of my presentation, I want to give you two examples of case studies we did. The first one is about the beta diversity networks and bacterial indicators of land use change. That was a European project where a field study with different sites across Europe. And the second study is this important of should we study a gram of soil, or do we get more information if we focus on smaller scales? So in the next slide, I introduce you to the bigger project here. And this project was about land use and land use change. And you see that we looked at 19 different sites across Europe and different land use types. This you see on the right side, you see a map of Europe. You see Scotland, Ireland was there, and different sites across Europe. Yeah, and you see the different colors present a different land use. So either cropland, grassland, or forest soils. And then at each site, there was also a conversion site. That means that directly next to that site, there was a change from the original site to another site. And that happened like 16 to 160 years before sampling. So for example, a grassland was converted to a cropland, or a forest was converted to a grassland or to a cropland. So then we did the physical-chemical analysis. And here we did something that is normally not done very often. We differentiated the carbon fractions. So we did not only quantify the total organic carbon, but we compared different pools of carbon. And I come back to that in a minute. And then we extracted DNA exactly with a kit like I have described, 500 milligrams of soil. Then we quantified the communities and differentiated the big kingdoms or domains, bacteria, archaea, and fungi. And then we sequenced the DNA. In this case, it was done with an already outdated sequencing method, but it doesn't matter. It gives the same kind of results. Pyrosequencing, 454 pyrosequences. And then we did multivariate statistics and network analysis. And you see in the next slide, you see this is a result now of all these 19 sites across Europe, and you can see that in the grassland, first of all, you see that the bacteria are the most abundant ones. So we use the copy number of the RNA genes, which occur in all microbial cells, as a means of their abundance. And you can see for bacteria, they are most abundant, and in grassland, there are more bacteria per gram of soil than in cropland and forest. And for the cropland, there are less fungi than, for example, in grassland. And in forest, there are less archaea than in cropland. Yeah, this shows you that even across Europe, you have significant effects of the land use type. And when now you look at the sequence diversity, there was a surprise for us that the highest diversity of bacteria we saw in cropland. Yeah, so that soil that is always mixed and that is badly treated, let's say, compared to a forest or grassland, but the diversity is higher here. Yeah, and then compared to grassland and forest. And we measured this diversity by using two different indices for diversity. And both indices, this is the Shannon diversity and the Simpson diversity, gave the same result, basically, that the diversity in croplands is highest than grassland and then forest soils. But the differences were more pronounced with the Shannon diversity. And when you look, what do the diversity indices, what are they based on? I don't show you the formula, but I just to indicate that the Shannon diversity is more strongly influenced by species richness and by rare taxa. So those do not occur very dominant, whereas the Simpson index is more about the evenness of the community. That means that if everything is at the same level, then the evenness is different as if you have very, very strong dominant and less dominant ones. So this means that the rare taxa appear to contribute strongly to the differences between land use, while the evenness seems less differential. The next slide. Now you see when you have the land use change here, then the communities also change. And you see that, and basically, to cut a long story short, the communities change to the direction of that land. For example, I told you that in cropland, there's the highest biodiversity. And if you convert grassland to cropland, the diversity increases. And actually, this was the only significant value we found. But the other values, even though not significant, they show the similar direction as the original land use. So the microbial communities adapt to the new conditions and evolve in their diversity towards the systems, towards the new systems, that we can say. So the next slide, we were able, and this is really, I should say that each site was most important, but we were even able to identify taxa across all Europe, so from Ireland to northern Italy and whatever, where we could say that in grassland we find more Gayella and Solirubrobacterales than in the other sites. And in cropland we find more Nitrospiras, Germanella and Bacilli than in other sites. And for forests, we were surprised. We found an enrichment of Rhizobiales and Actinomycetales. Yeah. So this is a very strong indicator, considering that we look at 17 different sites that have no relation to each other, except that they are all located in Europe. So now the question is, which factor filters the community, which is most important? Yeah. So this means we compare the community composition, and we find that the most important factor of a community is the site. Yeah, and that makes sense. So the sites in, let's say, in Scotland and in Ireland and in Italy and in Germany and wherever, each has a very particular community. Yeah, but and then we find, but also we find that the land use type has a factor of 10%. And we were disappointed at first to see that soil organic carbon only contributed by less than 1%. But now you move to the next slide, we see that now we take not the soil organic carbon as a sum parameter, but we use carbon fractions that we determine. So particulate organic matter and soil carbon associated with silt or with clay, etc. So different fractions. And then it explains 6%. So it becomes more important than the land use type. And the next slide, we take out the site now, the 25%, because that is boring to know. We know that the sites are important. So let's take it out. Let's pretend we take another one. And then we see that the pH and the soil organic carbon fractions, they are play 10% or 9% distinguish these communities. So if you look at the literature, everybody says pH is the most important factor for shaping a microbial community. And certainly it is a very important factor, but the soil organic carbon fractions are equally important. Only you don't see it in the literature because nobody measures this, because it's expensive and it's time consuming and it needs expert knowledge. Yeah. So if you read the literature, don't trust always just the data. Think about the importance of environmental factors that have not been determined commonly. Yeah, because I told you pH is always determined, but not the other. Now we can do network association networks. And we were interested in this soil organic carbon, which of them is important. And this means how are the taxa associated with the different soil organic carbon fractions? And you see these gray plots here, like POM is the particulate organic carbon, and SA is the silt-associated organic carbon, DOC is a dissolved organic carbon, and so forth. And then on the left, you see RSOC, this is the recalcitrant organic carbon. So you see the recalcitrant has no association at all with any taxon. And that makes sense because the recalcitrant organic carbon is not degraded. Yeah. Similarly, the dissolved organic carbon has only negative relationship. Why? Because the dissolved carbon is eaten up. So if there is a lot of dissolved carbon, there's no microbial activity, because that's what the microbes normally eat and use as an energy source. And the most links are with the particulate organic matter. And if we zoom into this tree, on the next slide, we can see now here you have this particulate organic matter, and then you see a very interesting structure. You see there are two groups that mutually exclude from each other, because the green lines, they show a positive correlation and the red line a negative correlation. And here is a big cluster of with Connexibacter, Actinomycetalis, etc. They are strongly associated with particulate organic matter. And then on the right side, you see this group of Propionibacteriaceae and Gayella, and they are not associated. They have a negative association. So if there is a lot of particulate organic matter, this group does not occur. Yeah. So this is interesting, but it is not true. Maybe we can go back 1 second, because my point here is that the green lines between each other, they may not mean a lot. It doesn't mean that OTU 53, for example, at the bottom, has a strong interaction with OTU 71, because they may just be at the same time there, but not interact. Yeah, it's like you have an increase of birth rate and the number of McDonald's places all over the world. And so there is a correlation, but it doesn't mean anything. So this is the same for these OTUs that correlate with each other. There is no, this is not a proof of interaction. So next slide, please. Here you see another example. This is the bradyrhizobium. OTU1 was the most dominant sequence we got, and it is associated also to particulate organic matter and silt-associated carbon. And then on the left, you see this recalcitrant soil organic carbon again, and it has no interaction with any taxon. So that is a nice proof that our network is meaningful. Next slide. Next slide, please. So the networks, they suggest interactions, but they only display associations, and they are poor indicators of interactions between taxa. So the question is now, how can we improve the likelihood of detecting meaningful correlations in soil microbial community analysis? And the answer lies for us in the spatial scale. That means now don't follow the kit, but extract DNA from less than a gram of soil. What happens when we reduce the amount for extracting total DNA? Do we still just see less of the same or increase heterogeneity? And I want to give you now a very strange example. I don't know if it works. I just made it up yesterday. So the question is the bacterial community composition seen in a 1 milligram sample the same as in the usual 500 milligram sample? And here you see something. Let's say this machine here, all the chewing gum in here, it has a distribution. It's a microbial community. And yeah, next. Yeah, no, just, yeah. So you see this is 500 milligrams. So you have the, no, go back, one step back. Yeah, so you have these 500 milligrams. And then you have a certain composition that I described above. Yeah, let's say 30% red. You can say for red, you can say bacillus, and for yellow, pseudomonas or whatever. It doesn't matter. It's just a composition. Now, when we take a small sample from that, yeah, that means a milligram. Do we get the same composition or do we get a different composition? If we get the same composition, the downscaling doesn't make any sense. If we get a different composition, maybe it means something, because we have a smaller size and the likelihood that we detect microorganisms which interact with each other increases. So next slide, please. So here just, this is stolen from a textbook. You see this structure, the aggregates. We have these macroaggregates, microaggregates, sub-microaggregates, and the primary particles I introduced you already to before. Now, the next slide. I made a rough calculation. It may also be wrong. I don't know. I tried my best. So you would say that, let's say in the macro aggregate, I calculated it may have a volume of 14 microliters. and the weight of 22 milligrams. And a micro aggregate would be 1,000 times less. So where are we in the extraction? In the extraction of DNA, you see the 250 milligram sample, we are way above macro aggregate level. That would mean like we have 100 of macro aggregates together. Yeah. Whereas when we extract from 1 milligram of soil, we would have something between a macro aggregate and a micro aggregate, maybe individual macro aggregates. So from my group, Marton Soboschlei, he made some experiments. And this is one. He took 250 milligram soil samples. He did the usual extraction, but then from the homogenized slurry, he took samples that represent 1 milligram of soil, 5 milligram of soil, and 25 milligrams of soil. And they all should be the same, because we were worried that if we take too little template DNA, we get a PCR artifact. Yeah. So also we included 6 controls here without adding any DNA to the kit. And the result was surprising. You can see so the 16S genes were not only detected in the samples where we had the soil, but also in the kits without soil DNA. So these kits, they are contaminated with bacterial DNA, but you can very well identify them, and then you can look at your community with the soil, and then you see that these contaminants disappear. Or if you want to be on the safe side, you just kick out the contaminants from your further analysis. And but the low DNA from the homogenized soil slurries gave highly similar results. Yeah, and low DNA from small sizes, however, were less similar. So in the next slide, you can see that more graphically, or no, the very afterwards. So the next thing we did is we extracted 250 milligrams of soil, and then we single aggregates. This is the one where we have this frame around that is single aggregates, just to, and then we quantified and sequenced these samples. And to the next slide, you see that for the qPCR, this is a kit we use. And you see that the 250 milligram sample, if we reduce the amount of DNA, we get even a higher yield. Yeah, so the kit, which is supposed to be the best designed for 250 milligrams of soil, worked best with 25 milligrams of soil. But it was, the efficiency went down then with 5 milligram and 1 milligram of soil. Still, it was all in the same range. So we were able to very well quantify bacterial communities also from 1 milligram of soil DNA. And so DNA extracted from 1 milligram of soil. And then we look at the aggregates, we see the same. We see that we were able to extract a lot of or detect bacterial communities in aggregates similar to the amount we detect in the common 250 milligram sample. So the next slide. Here you see now the principal component analysis, and what it tells you is that if the dots fall into the same spot here, they mean they are identical. And if they scatter around, it means that the community composition is different. And you see a very nice result here. You see that the 250 milligram, these are the black dots, and you're surprised you only see one dot. It's because they all fall exactly into the same spot. So they are really like replicates. This is from the same soil, yeah, from the same bag of soils. And the same for the 125 milligram. But if you reduce further, you see that now with 5 milligrams of soil, and even more with 1 milligram of soil, you get different bacterial community compositions. And the same you see when you compare aggregates. They are also these yellow points. You see they are very, very different from, or more heterogeneous than from the 250 milligram soil samples. Yeah. The next slide. This is a one I have to take you through a little bit. So what do we see here? We see from top to the bottom, we see the amount of soil from which the DNA was extracted. And then you see from 1 to 8, these are the bacterial sequences you find in 8 replicates, or in 7 out of 8 replicates, or in 6 out of 8 replicates, and so forth. And if you look at the bottom, the 250 milligram soil, you see you detect 208 sequence variants, and they occur in all 8 replicates. Yeah, you find no other sequences, and the same for the 125 milligram of soil. You detect 214 sequence variants, and they all, except for one single variant, they occur in all 8 replicates. Now, if you move up, you see a surprise. It changes completely with 5 milligram and even more with 1 milligram of soil. So in 1 milligram of soil, you detect also as many, you detect 199 sequence variants. It's almost as much as in the 250 milligram sample. But you see a number of samples that occur in 7 out of 8 replicates, in 6 out of 8, and so forth. And to go to the right, you see in 1 replicate out of 8, you see 100 sequence variants. That means, and when you see at the composition of them, they look different. Like the gray ones, these are in this scale Taumarchiota, they did not occur at all. But there's enrichment of the blue ones, and that's like the plantomycetes, which were the ones that love to be close to clay, right? So they are they seem to be scattered. They occur in some aggregates, but not in others. Okay, next slide. And when you do a network, with the 250 milligram size samples from the same soil, you cannot get a network because the replicates are all the same. You cannot make a network if everything is the same. But if you take 35 replicates, so 35 individual aggregates from this soil, you get a nice network of organisms. And if you see on the next slide, you can even distinguish 2 groups here. And this is one group is this Taumarchiota actinobacteria cluster, and the other Acidobacteria cluster. And they interact with each other strongly, but they are linked by 3 keystone taxa. Yeah, and I don't want to go into too much detail here, but it shows you that if you go to the small aggregate levels, you can see, in fact, interacting communities that you cannot see with the common methods. So just at the end, what we see is an aggregate, and Eduardo, if you press once again, you see a highly sophisticated animation. Yeah, this is it. So it shows that there is a metabolic turnover. We see that an aggregate receives energy and carbon, and then it transforms it. Of course, a certain fraction is lost by heat, but then metabolites are created, and this process is controlled by many different factors. And we believe this is a nice approach for the future to study soil microbiology, to understand interactions and how communities are working with each other and changing the environment. Next slide, please. The next one. So my conclusions, and I have 3 conclusion slides, so sorry about that. For the geographical scale study, I just say each site is characterized by a distinct community, which responds to land use in a similar pattern independent of the location. Soil organic carbon fractions have a strong impact, but it is rarely measured. The impact is equally to pH. And across European sites, this was quite surprising, there is a dominant bradyrhizobium. This is the most dominant representative. And we could find some indicators for land use independent of the country and location. And the next one, for the scale, so as the particle surfaces, I could show you they have a preference or avoidance of the specific bacterial and fungal taxa, and thereby they structure the community. You can extract DNA from as little as 1 milligram and unveil hidden heterogeneity and co-occurrence networks of soil bacterial communities. And I think more effort should be put into downsizing DNA extractions to detect functional units and interacting partners. The next slide is just like recommendations. Do not select only explanatory variables of soil only because they are easy to measure, but select them based on scientific needs. Yeah. First define the objective and then choose the amount of soil you want to look at. not just trust the company who tells you should use 120 or 250 milligram. Network analysis can be good, but they should be carefully if you overinterpret them, because it may co-occurrence does not mean interaction. And the next one, the likelihood to detect truly interacting partner correlates negatively with the size. So the smaller the size of the soil, the higher is the likelihood of interactions. And network analysis based on 250 milligram of soil or more should only carefully be interpreted. And I think this is my last slide. So before I finish, I want to thank the people in my group: Martin Soboschlei, Sainur Samad, Damini, Beilu, Wan Liangzhu, Karin, Tresha, Britta Müller, and Jana Ozarek, and my collaborators from Thünen Institute of Climate, Smart Agriculture, Christopher Pöppler and Axel Don. And here's a picture, actually. It may not look spectacular, but for me it's spectacular because it's the view when you land in Linate, in Milan Linate, and I miss it so much to go there because I enjoy so much being in Emilia-Romagna and this region. And so this, hopefully I can have that view in the future again, and also to visit, of course, Eduardo, and for further collaboration. And with this, I want to thank you for your attention. Thanks. And I take questions, of course.
Is muted. Okay, so here we are. Thanks a lot, Christoph, for the talk. That's really insightful, very, very interesting, for sure. I was quite astonished to see when you showed the data about the diversity in the pastures and cultivated sites. Actually, with Piersandro Cocconcelli and Marco Trevisan, we saw something similar in an Italian site in the same year. So it was quite similar outcome. That was very interesting. So I would like to give of course, the time for question. We have quite some time for questions. So I would like, first of all, the school attendant, but since it's also a public event, anybody who has got a question for Christoph, please go on. And I would also propose you to use the hand. You know, there is the raise your hand button here on Teams. So if you can use it, you can go for a question. So let's see if something is arriving. Well, I will start with one, then we will have other ones for sure. So this thing about the size selection indeed is quite astonishing. I knew a little bit, but not. I didn't thought that the effect were so big. So at the end, you gave this advice that depending on the kind of studies and objectives, you can change the size of samples that you're extracting, right? But are you already, I don't know, imagining, I won't say some kind of rules, but in which cases is it more appropriate to use a small size and which you can use 250 or 500 milligrams?
Yeah, so first of all, this is also very good news, basically for the kids, is that when you take the recommended amount, you get really replicates. Yeah. So let's say you go to a field, you take, because you don't sample 1 gram of soil, right? You sample a whole bag full of soil. Let's say you sample a kilogram of soil. Then you go to your lab, you sieve the soil, and then you take an aliquot of that and you extract it. And that means when you extract 500 milligrams of that bag of soil, you pretty much have a representative number. So that's a good news for this particular soil. But we have now, in the meantime, we have done it also with other soils. But if you want to study interactions, yeah, let's say you have a soil and you want to know who is interacting in that soil, then you cannot do it by 250 milligrams sample because you always get the same and the same and the same. But if you look at the individual aggregates, you get variation in this pattern, and then you can generate networks. And then you can say, okay, in this soil, there's a group of Traumaceota that has a strong link to a group of acidobacteria. And, so I would say always when you want to study interactions, and let's say that could be sharing metabolites or something, or degrading pesticide or whatever, then I would recommend to go to the very small scale. But if you want to study, let's say, in an agricultural field trial, you want to study the effect of different fertilizers, then you would go at a higher scale, yeah? Or if you want to study a geographical variation, then you extract 250 milligram or 500 milligrams of soil.
It's okay.
But you are right. We should define rules. It's not, I mean, I would not make the rules, but I want to discuss them, and this could be a way forward to go. And my big complaint is that we have so many people working with soil DNA and nobody cares about that. And I don't understand it. And so hopefully we can fuel a discussion here in the future that people should be more aware and think, is this the best way? Is this the right amount to extract DNA? Yeah.
Okay. So let's see questions from the people, from the public. Come on, especially from the students of the school, because come on. I don't believe you are so shy. So you can use the raise hand. Okay, there is Serrani Dominique. You can, of course, when you make the question, just turn on your camera and your microphone, nice. So we see each other. Okay. So Dominique, please. I saw you are raising your hand.
Probably has to open the microphone. I don't know.
Microphone and camera. Yeah.
Okay, sorry.
Okay. Hi. Hi. Hi.
Sorry, wait.
Please.
I hear you well. Not anymore.
We don't hear you, but check at the beginning we were hearing you.
Okay, right nice. Now? Yeah.
Perfect.
I'm so sorry. The computer is very slow.
The
connection is not good. Sorry. Good evening to everyone. I'm Serrani Dominique, a PhD student from the Soil Science Department in Ancona. I want to say thank you to you for the presentation. It was so interesting. I have a question about the method of soil sampling. How, in your case studies, was done the soil sampling? Within, I don't know, 30 centimeters, they utilized horizons to take sample? Is the question, the method?
Yeah, so for the length, so if you have sampled, first of all, if you, let's say you sample from a cropland or arable soil, we always, we remove the first 1 or 2 centimeters because there is an impact of radiation, of sunlight, etc. But then we assume that the plow layer is mixed all the time. So there from cropland, we take from -2 centimeter to -6 or 8 or 10 centimeters normally. So we regard this as homogeneous. For forest, it's a bit more complicated. Normally what we do is we remove the litter layer, the organic layer, and then we take the mineral, the first 5 centimeter depth from the mineral, from the real soil. And for grassland, there's another problem there. Of course, you have so many roots. You always have this strong rhizosphere impact that you cannot separate. So I would say for each land use type, you need to have a specific sampling strategy. But there is no general rule to that. Yeah, and it gets really complicated now, like in our European study, when you compare the samples with each other. In there, we really remove the upper surface area, not to have this impact, for example, forest from the leaf litters, or we try to remove all the roots from the grassland and then extract the soil. Yeah, but of course, for grassland, it's not possible to. I think in grassland, anything is rhizosphere soil. It's very difficult to have something else.
Oh, thank you so much.
Yeah, you're welcome.
Okay, thanks, Dominique. You can maybe turn down your hand virtually, okay? And so we can see if there are other questions from people around. I will go.
There's one.
In the meantime, there's one. Okay, Paola, please. The president of our society.
Okay, yes. Thanks, Christoph. Very, very interesting speech. Very, very interesting data and a lot of critical thinking. I'm very interested to the preferential association of bacteria species with different particle size fractions in soils. Are you assuming that are single particles? The first question, because you use a mild sonication, so maybe are not single particles of silt or sand, but are aggregates. Yeah. And second question is, did you analyze the mineralogy of these different particle size fractions? Is curious for me. I'm curious to understand why a bacteria select a clay particle, maybe a kylonite or a smectite, or another species select a feldspar, for example. I don't know if it is a sand fraction, you know. Why do you think they have this preferential association?
Yeah, so first of all, the method, it's you are absolutely right. If you reduce the ultrasonication too much, you cannot separate the particles from each other, but we calibrated it. So we work with the soil from a long-term field study that is very well known, and we calibrated it with so we used a conventional protocol with the full ultrasonication that was published. It's a method by Amelung, published in 1998. And then we reduced, and then we saw that we still got the same percentage or a similar percentage. I have to admit that for the clay fraction, our numbers were always slightly higher, considering that probably it is not as perfect separation as with the very high ultrasonication energy. But still, it was good enough. It was only a very minor percent differences. The reason why there are different preferences is very clear, because the mineralogy of a clay particle, and maybe you know that even much better than me, is different from that of a silt, of a fine silt or coarse silt, or sand. Like a sand grain is silicium oxide, has totally different sorptive capacities than a montmorillonite or whatever. So, and this is also a collaboration with Ben Christensen from Denmark, who was one of the popes of this primary particle fractions, who got really excited about our data, and he said, so he confirmed, he said that there's the different particle size fraction represent totally different types of surface areas, and that means that the compounds more easily or more difficulty adsorb or desorb, and these compounds are used as energy source. So probably the microorganisms, for example, that are associated with clay need more energy to get a compound than at a silt particle, but that is only speculation. What is also interesting is that we always studied not only the soils but we compared soils with different, from long-term fertilization, so which had different amounts of soil organic carbon, and these long-term fertilizations had only a very minor effect on the preference of the taxa to the particle size fractions. So it is really something that has to do with the mineralogy.
Oh, right. Very, very interesting. Thank you very much.
Thanks.
Okay. Do we have other questions? Let me see. I think there is a point. Enrica Picariello, is it correct? Please.
Yes. Good
okay.
afternoon.
If you have got also the camera and you're not shy, you can also. Yeah, or just gone with the question, as a.
Sorry, but sorry. I have to. Yeah. Okay. Can you hear me now?
Yeah.
Yes.
Yes.
Yeah.
Please.
Congratulations for the presentation. Very interesting presentation. I'm Enrica Picariello from University of Sannio, Benevento. And I have just two questions. The first one is, how do you explain the lower bacterial diversity in forest soil? And the second one is, why the bacterial diversity change just to land use change when land use change from cropland, from grassland to cropland?
I'm not sure I understand the second question, but the first question, why is the bacterial diversity in forest soils smaller than in cropland soil? Let me answer that first, and then you can ask.
Sorry, the second one is,
how
do you explain? Yeah, how do you explain the fact the bacterial diversity change after just when the land use change from grassland to cropland?
Yes. So the first, why is the bacterial diversity in forests lower? So these why questions, they are kind of, you ask an old guy like me where so I don't have the data for that. So all what you ask here is my personal opinion or my hypothesis. And so my hypothesis for the forest, why is it so low, is because I think that the forest soils, the contribution of fungal mycelia is much stronger than of bacterial diversity because it's very structured, and that probably the mycosphere, that is the surface of the mycelia, has a strong influence on the bacterial diversity, and maybe that means that most bacteria that you see are associated with fungal mycelia, and then maybe there are less different niches. That could be one reason. For the cropland, I think the diversity is very high because a lot happens to the soil. So the farmer adds fertilizers, he mixes the soils, straw is decomposed, plant roots remain on the field. There's a lot of mixing, and this always promotes the growth of bacteria. So there are more substrates available. So I assume that in cropland soils you have more R strategies. So bacteria that degrade very fast and become dominant. That could be maybe even tested in the future. If you, for example, we know alpha proteobacteria, so some groups or bacilli, they respond to these conditions. Okay. And when you have a land use change, then and for grassland the same. You have many roots and a lot of contribution of the fungi. And we didn't measure the fungal diversity in the systems or the fungal mycelia. So I think that's the reason why the importance of fungi in grassland and in forest is more stronger than the importance in cropland. Okay. I hope this answers a little bit your question.
Perfect. Thank you. Very interesting. Thank you.
Thank you for your question.
Okay, I think there is a last question, but it was written in the chat line by Stefano Moccali. Probably he was right, so I can read this question. Or you wanna talk, Stefano? Are you in?
Yeah, I can also talk. Yeah.
Do it directly. I feel it's better.
Thank you. I don't have the hand icon on my screen, so I prefer to. So hi, Christoph. First of all,
hi, Stefano.
Nice to hear you. Your great presentation. I really enjoyed. Thanks. I've got a question regarding the soil sampling, because you touch a very, very critical issue regarding soil sampling. It's a really complex topic, and I really appreciated your message, your invitation to consider the size of the soil sampling depending on what you are looking for, right? So I just wonder to which extent the soil composition, especially regarding the soil organic matter, soil organic carbon fractions, should eventually taken into account when you plan your sampling in the field. So I don't know if the distribution of soil organic matter could consistently affect the kind of soil sampling you have to plan. So your opinion about this.
Okay, yeah, I think for the soil organic matter, there are different pools. There is a pool that is non-microbiologically active. That is what we call the recalcitrant soil organic matter. People used to call that, what is it called? I don't know, some kind of non-degradable humic acids. Now the new concept is a bit different, and but you have different fractions. For example, particulate soil organic matter has a high turnover, and the smaller the particle sizes, the less accessible is the soil organic matter. For example, clay-associated carbon is less biologically active than silt-associated carbon, and so forth. For the sampling, I don't know exactly what you mean, but maybe, for example, the seasonal impact is, of course, very strong. When you sample a soil from the field, you should be very careful when to sample it. And normally the common soil microbiological recommendation is to do it at the end of the winter season, because then you have equilibrium conditions. Mainly, you don't have a fresh input of organic carbon by roots and by rhizospheres, and then you have more stable conditions. Some other people say it's good to sample after crop harvest, let's say in September or October, but it depends also on the region and also your questions. But certainly some fractions of soil organic carbon are highly influential. Yeah? And the most is, of course, the rhizosphere, but it's also the most, it's immediately metabolized. So it depends. I don't know.
Yeah. I just wondering if when you have to select the kit, the commercial kit for DNA extraction, how should you consider eventually the soil organic content of the soil sampling? So that was the question. So,
I don't know. Yeah, it depends on the project. I don't know. Maybe. Of course.
Because it's really interesting. That's really complex to me.
yeah, absolutely.
Challenging.
Okay.
Thanks a lot.
So maybe I see a last question. If you are available, Christoph, then we can close. From Flavio Fornasier. I read it on the chat, but if you want to take the microphone.
Can you hear me?
Yes.
Okay, thank you very much. I thank you very much, Christoph, for the very interesting speach. And the question is, basically, I am an enzymologist, and I usually perform multi-enzymatic tests simultaneously. I'm really interested in biodiversity. So the question is, you detect an increase of biodiversity, let's say from forest, changing land use from forest to grassland or another one, okay. But this new microbes comes from what place? Were they already in that specific site, so we're not able to detect them due to intrinsic limitation of the methodology, the extraction, or they are really imported because we know that spontaneous generation of new species does not exist. Thank you.
Yeah, that's true. That's what we know, but the rest we don't know. And I think everything, yeah, I mean, mainly it's a selection process. So let's say when we sequence 2,000 taxa from a forest soil and then 2,000 from a neighboring grassland, then we see some very rare that did not occur before, but we don't see, let's say, like very dominant groups that have not been there, at least in lower numbers before. So I think it's a modification. So the main factor, and you should imagine that these bacteria are at these individual sites for not for 100 years and not for 1,000 years, but for millions of years. Yeah, the soil was there already many million years ago. And the communities in there, they have a long, long evolution. So when you disturb the system, I am pretty sure it does not open a totally new niche for a totally new imported microbial community, but it only selects for specific members that were less abundant and that become more dominant then. Because land use change is dramatic, and when you think about deforestation, it's terrible what happens when you think about the Amazon or everything, but even in Europe, it may be a modification, but the major community contributing is there for millions of years. So I don't think it is important. The main new organisms are imported from somewhere else.
Okay. So basically, I think we can close the session. We had a lot of questions. So thanks a lot, Christoph, really for being so kind and available. And basically, well, now we close this public session. We are moving now to the private session, let's say private, which is for the school participants. And of course, Christoph, if you want to join in the next days, if you're available, like tomorrow there is a Stefano keynote, another presentation, we will send you the invitation to join.
I would love to. I have another appointment though, but maybe you can send me. Maybe I can get rid of that appointment because I'm really interested.
Okay.
But I'm not sure I can make it. Yeah.
We will send you the Teams invitation, and you are free to decide to join or not.
Thank you very much.
But you can do like our friend Pier Sandro and open for 4 teams altogether. Yeah.
So I wish you all a very, very successful school and enjoy as much as you can. So thank you very much. And
Thanks.
yeah, bye-bye. Bye.
It's been a pleasure and an honor to have you here. Thank you.